Hello, my name is Rebecca Shelley, and today I will present my work on the role of mutant KRAS in the loss of PP2AB56 alpha in the transformation of pancreatic epithelial cells and the progression of pancreatic cancer. Pancreatic cancer is the fourth leading cause of cancer-related deaths and has a 5-year survival rate of only 10%. KRAS hyperactivating mutations, like KRAS G12D, are extremely common in PDAC and promote downstream KRAS signaling and oncogenesis. As of right now, KRAS remains a difficult target for therapeutic intervention. PP2A has been found to suppress many downstream effectors of KRAS, and PP2A inhibitors are overexpressed in PDAC cell lines, suggesting that the suppression of PP2A contributes to oncogenic signaling. PP2A consists of 3 distinct subunits. While the A and C subunits only have 2 variants, there are an abundance of B subunits as it controls substrate recognition. B56 alpha has shown the highest tumor suppressive capacity of PP2A B subunits. It has been found that B56 alpha is mutated in approximately only 0.4% of overall patient samples with PP2A mutations. This low rate of mutation allows the opportunity to therapeutically target the PP2A complex. The goal of our research is to determine if the loss of PP2A B56 alpha and the expression of its inhibitors increases in and accelerates early PDAC lesion formation, and to identify how oncogenic KRAS suppresses PP2A B56 alpha activity. Both in vitro and in vivo studies look to determine the cooperation of the loss of PP2A B56 alpha with oncogenic KRAS to transform cells or enhance the progression of pancreatic cancer. In a panel of PDAC cell lines, PP2A activity is decreased while the expression of its inhibitors SET and CYP2A is increased compared to normal pancreatic epithelial cells. Decreased expression of PP2A B56 alpha is associated with decreased disease-free survival, and CYP2A staining shows an increase in CYP2A expression as tissues become more abnormal, which indicates that PP2A activity is likely suppressed by the expression of its inhibitors. We created a KCB hypomorph model with the loss of PP2AB56 alpha to compare with KC mice, both with KRAS G12D mutations in the pancreas. Isolated acinar cells from the KC and KCB mice were cultured ex vivo on collagen. The results indicate that the loss of B56 alpha accelerates the acinar to ductal process, as the KCB mice have a higher number of ductal structures compared to the KC mice, and show increased expression of ductal identity markers by day 3. At both 2 and 5 months, H&E staining in KCB mice show further progression of pannin lesion formation than in KC mice. Alcyon blue staining, used to mark pannin areas, indicates that KCB mice have significantly lower healthy acinar area and have a higher number of pannin lesions per area than KC mice. Together, these results indicate that the loss of B56 alpha can accelerate PDAC initiation and progression when coupled with KRAS G12D mutations in vivo. To determine if the loss of B56 alpha can transform epithelial cells when coupled with KRAS mutations, we are generating stable cell lines from HPDE cells that have an inducible expression of KRAS. These cells will be virally transduced for knockdown of PP2A B56 alpha. Soft agar, clonogenic, and proliferation assays will be used to assess if the cells have been transformed, and Western blotting and qPCR will be used to determine signaling changes in the downstream effectors of KRAS. To determine if the loss of B56 alpha with KRAS expression enhances PDAC progression, we will characterize differences in tumor progression in 2 and 5 month tissues from KC and KCB mice. Trichrome staining of 5-month pancreatic tissues shows that the KCB mice have a higher amount of collagen deposition than KC mice. Further stains will be used to characterize morphology, progression, and expression of PP2A inhibitors within these tissues. Future directions of this project include using HPDEI wild-type and iKRAS SHB56 alpha cell lines to determine signaling and phenotypic changes, and continuing tissue characterization from the KC and KCB mice through different tissue stains, along with using both cyclic immunofluorescence and immunohistochemistry to look at the expression of B56 alpha inhibitors and their localization within the cell. In summary, PDAC cell lines have increased B56 alpha inhibitor expression and decreased B56 alpha activity, showing that PP2A B56 alpha suppression plays a role in oncogenic signaling. Loss of PP2A B56 alpha correlates to poor prognosis, accelerates ADM, and accelerates tumor initiation. Finally, we intend to understand the mechanisms of the cooperation between oncogenic KRAS and the loss of BP2A B56 alpha in transforming pancreatic epithelial cells and progressing pancreatic cancer.